from Microbe TV. This is office hours for Wednesday, July 30th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can learn some virology and maybe some other sciences too, ask questions, and you won't get any lies or misinformation. Tonight we have some news items. We're back to the news, unfortunately, and a mini lecture, part 2 of reverse transcription and integration. Before we get going here, people are asking, what are these crowns with the number 1, 2, and 3? So this is a recent YouTube thing to show the top chatters. So if you chat a lot, you get number 1 or 2 or 3. And obviously it's a dynamic thing because quiet people can start chatting a lot and they can grab the number 1 crown. Okay? It's a crappy looking crown. It's not very good. Anyway, tonight we have our moderators, Vanity Nutrition. Thank you for joining us. Les Fabi. We have Steph SF. And we have Tom Steinberg. Thank you all for joining and helping to make this a civilized chat. Tell us where you're from. We'll shout you out. And then we'll get to some news. By the way, I was in Brooklyn today. Oh my gosh, it was 34°C, bright sun, not a cloud in the sky. Whew. Pretty intense. And can I show a picture? I will. Let's see if I can get this tech up quickly. And what we were doing in Brooklyn is checking out the Gowanus Canal. The Gowanus was the subject of an episode of TWIM. I don't know, some time ago. I forgot the number. Here it is. Here's a pic I'd like to show you, and you can tell how hot it is. Let's make that a little smaller. Oh my gosh. So it's the Gowanus Canal behind me, and it starts in the lower bay of Manhattan, kind of on the other side of the Statue of Liberty, on the New York side, south of the Battery in Brooklyn, and it winds around a bit. So here it is, and this is all kinds of industrial stuff going on here. There may be other parts that are calmer. But these industries are just continuing to put stuff in the water. It was very interesting. Anyway, we put up a little video today on the socials, very short. It was a beautiful heron standing on the banks. And tomorrow we'll have a longer video, just a minute or two. Because I just wanted to see it, what it was like. Now, I would really like to walk the whole Gowanus. It's too hot today. Too hot. And I'll go back in the fall when it's cooler and walk the entire length. I want to see what it's like. One of the cool things we found, and I was there with Kim, Microbe TV's social media assistant. They have a nice little garden on the side. They're developing some parts of the sides on the Gowanus. And the lady who's taking care of it today, she let us in because it's normally closed today. And we walked through it. So they got nice plants. It's open on certain days of the week. You can go in there. And we got pretty close to the water. It didn't smell a lot, but there may be other parts that smell. Anyway, let's see. Who's here? Wayne is from Urbana, where it's 24°C. Tom is in the Oregon Coast Range. Welcome back. Steph is in San Francisco, where it's 62F and windy. Unusually cold July. Well, it's been really hot here in the Northeast. It's supposed to get a huge thunderstorm tomorrow. Lots of flooding. So I don't know if any of you saw, but a couple of weeks ago when we had this horrible storm in the Northeast, they were showing videos of water pouring into the subway. There was First number 1 train, which is 7th Avenue and 28th Street Station. That's right near the incubator. It was funny. I go into that station all the time. And Vanity Nutrition is here from Long Island. Viruses Luna is in Iowa. Hello, Viruses Luna. John is in Minneapolis. 25C. Visto is in Sydney. 12C. Well, it's winter down there, right? Dean is in a Smoky, Wisconsin at 24C. Leo is in Mendocino, 56F, with some fog. Leo has a fantastic sunset picture. Kang is in Chicago, 20C. Another storm coming. Vincent, you got to come to Iowa. Hope so. Hope I can. I was in Montreal 2 weeks ago. That was lovely. And what trips do I have planned? I'm going to Thessaloniki, Greece. In September, I'm going to be at the Harvard Virology program retreat in September. Going to Virginia Tech, I think, in October. December, Germany for the Giant Viruses meeting, and a bunch of stuff next year. Oh, I'm going in May to Corvallis, Oregon University. Noir, welcome. I'll be chasing the lightning after 9 p.m. Dramatic skies tonight outside with the tripod. Hope I don't have a Benjamin Franklin moment. In Santa Fe, New Mexico, 78F. Monsoon season. I see at the top it says 75 XP. At the top of what? I don't know. I don't know where you see that. I don't know what it means anyway. Jeff is in Elgin, Illinois. It's 23C and not raining. So Vanity explains why she is still in Long Island instead of Montana. They expected seats to open for me and everyone else accepted pending seat. They did not open. I was, however, offered a seat in the class that starts next year. Okay. Better than not at all, right? And there you go. The crowns are related to chat engagement. Engage a lot. Hello, Crystal in Edmonton, Alberta. Welcome. Brian is from Montreal. Sorry you couldn't get together. Brian, my trip was abbreviated, as you know, because of the storms. Steph doesn't like the crowns. No, I don't think they add anything. I mean, I don't think any of you are going to chat more to see a crown next to your name, right? No. EHS Talent is in Lehigh Valley, Pennsylvania, 28C. Air quality alert. Look in the top right corner of the chat. Click on that crown. Let's see. It says top fans. Oh, look. It says 1, 2, and 3. And then it tells everyone else all the way down to 28. Wow. That's cool. Thank you very much for that. That was Wayne who told me to do that on the stream. Carol, our favorite nephrologist, Bakersfield, California, where it's 36C. Jeff says, I raise monarch butterflies and have had a miserable year to date. More have suffered from what appears to be nuclear polyhedrosis virus infections. Could hot weather make this worse? I'm not aware of it, but the dehydration could couple with infection to make it worse. Laura is in Washington, Pennsylvania, where it's 30C. Yeah, Visto thought it meant that he's second cousin to the king, but you've already lost number 2, Visto. It's very dynamic. Very dynamic. Crystal is in Edmonton, 26C, not smoky today. Betty K is in Washington Heights, down to 25C. Logotic 22C and a thunderstorm brewing in Chicago. SRR is in Paso. Well, this is the way they say it in California, Paso Robles, but I like to say Paso Robles. 82F. Pete is in London, back from a hot Dubrovnik, and Kotor in Montenegro. So Dubrovnik is where European Congress of Virology meeting was earlier this year, which I sent Rich Condit to, and we recorded a TWIV from it. Very interesting TWIV. Looked like an interesting city. Sorry I couldn't make it. Area ozone is in Asheville, 25C. Mike and Lori Dinger in the Great Northwest, 27C. Soul Symphony. Hello. Privyet Elena from Reno, Nevada. Surprisingly mild summer, 24C at 5 p.m. at the end of July. Not complaining. Guess all the heat is on your side of the continent this year. Doug is in Redondo Beach, where it is 73F and the tsunami warning has been canceled. In Iowa, where virus's Luna is, it's 26C, and Lulu is having a catnip spa. Thank you, SRR, for your support of science communication, donating through YouTube to raise the algorithm. Jeff says, the coldest winter I experienced was summer in San Francisco. I visited San Francisco. It was very cold every day, but it did get up to 75 one day. Walnut Creek was far warmer. Hello, Questella. Are you enjoying your books? I hope so. So Wayne says, click on that 75 XP. For me, it's a crown that says top fans. There's no 75 XP. So I assume that you're... the graphics aren't coming through for you, but it's no big deal. Jeff says San Diego at night was in the 50s. I thought California was warm. Didn't bring anything warm. Luckily, my friend had extra sweatshirts. Martha, hello from Charleston, where the heat wave has moved on. Vanity says, if we posted for the sake of crowns, our chat would be less organic. No thanks. We can leave that for the Rogan, etc. clouds. Well said. Very well said. Hello, Kip. Hello, Vincent. During these current social debates fights, the craziest people are the loudest. Well, we know who the loudest ones are. Rob, thank you for your support of science communication. Vincent rules. I don't know if I rule anything. I just try and teach you guys in the world. That's what I do. College Kid is in Provo, where it is 97 degrees. Hello, College Kid. Kip, thank you for your contribution to science communication, your allowance. Cool. Yes. Questella says yes. I said, are you enjoying the books? She said yes. Okay, you can be terse. It's fine. John says hello from Freedom, Maryland, where ticks carry anaplasmosis in my yard. Okay, folks. Let's do some news here. Okay, so told you about the goannas. My visit, so check out YouTube or the socials tomorrow. You'll see some more video from that. Okay, first of all, a story from NPR, and let's share it so you can see it. In a first, Senate confirms a new CDC director. Okay, so what's the first is that in 2023, a law was passed saying that CDC directors from now going forward are not just appointed, but they have to be confirmed by Senate. And the vote was 51 to 47 along party lines. So that should tell you that there is possibly something wrong with Susan Monterez if all the Democrats are voting against her. But I don't know her. She's apparently got a long experience in public health. So she's a microbiologist, and sorry about these ads, a microbiologist and immunologist by training. She served as the acting director from January through March and then stepped down when she was nominated. She was previously deputy director for the Advanced Research Projects Agency for Health. Okay, so remember, Dave Weldon, this Florida physician, completely unqualified, was originally nominated. And the Senate reached out to Trump and said, we don't have the votes for Weldon. Now, why didn't they do that for RFK Jr.? Right? Not the right guy for the job. So what about Monterez? This is what bothers me about Monterez. One of the things. So she was in a June confirmation hearing. She said, restoring trust in CDC was one of her top priorities. Okay. I will rebuild credibility by making CDC leadership more public-facing and accountable and ensure all recommendations are backed by publicly available gold standard science. I hate this friggin' term gold standard. This is crap that RFK and HHS have brought in, and it's just nonsense. But Bhattacharya always talks about gold standard science. This is what we do. We always use gold standard science. So don't think you're inventing anything novel. So this gives me a lot of pause. Now, maybe she's just sucking up to the administration because she wants to get confirmed. It could be. But that and the fact that the vote was along party lines really bothers me. All right. Now, what else is she going to do? There was another line here I wanted to tell you about. She had a question from Senator Lisa Blunt, a Delaware Democrat. Here's where I'm reading. About whether she would commit to keeping routine vaccines free for U.S. children. And she said, vaccines absolutely save lives, and if I'm confirmed as CDC director, I will commit to making sure that we continue to prioritize vaccine availability. Okay. Fine. You're committed to vaccines. Now, how does this jive with RFK Jr.? Well, it doesn't. She also believes that fluoride is an important component to oral health and that the fluoridated tap water in her community is safe. Now, Kennedy, of course, wants the CDC to change its guidance on fluoride in water. So let's see how that works out, right? Are they going to clash over it? And after hearing, Monterez repeatedly defended the Trump administration's vision for CDC, which she said is getting back to its core mission of preventing, detecting, and responding to infectious diseases and emerging threats. She said programs that focus on other health issues like chronic diseases, obesity, depression, drug addiction, and overdose would be transitioned to other parts of HHS. So she says this is getting back to its core mission. But actually, it's been CDC's mission for a long time because it's called the Centers for Disease Control and Prevention. It doesn't say Centers for Infectious Disease Control and Prevention, okay? So I don't buy this at all, getting to the core mission. I think that these other diseases are perfectly within the purview of CDC. So anyway, she praises the secretary who has laid out a very clear vision for making America healthy again. Really? He's laid out a clear vision? Eliminating vaccines? Really? Is that it? I think this is just terrible, okay? I hope she's better than this. Gold standard. All right, enough of this. No, this is just annoying me. You get the idea. Okay, what's next? Oh, this is an interesting. This is a dumb article. Oh, I have John up here the whole time. I'm really sorry. Okay, this is perfect from Molly, who is in Hilton Head. It's 101. How can we get rid of chronic disease if we all die young? We can get rid of chronic disease if we all die young from infectious diseases. Yes, thank you for pointing out the ridiculousness of his plan. And there is no plan as far as I know. He thinks that chronic diseases are all caused by vaccines and that he's going to eliminate vaccines and that's it for chronic diseases. This is so stupid. It's just stupid. It's following one person's zealotry. Science does not work that way. Look, the Soviet Union followed one person's zealotry, Lysenko, and as a result, many people died of starvation. Should know better. This is ridiculous. Anyway, the next, we'll take Molly off here. Okay. The next article, oh my gosh. And this is in the City Journal. I don't know what that is. Was COVID a Chinese bioweapons research project by Judith Miller? A new report from a weapons of mass destruction expert raises disturbing questions. Okay, so this is a separate report that you may have seen, which says the pandemic was probably the result of a military research-related accident in a Chinese lab. And the work may have been done in violation of a treaty banning biological weapons. All right, where's the evidence? Okay, there's no evidence for it at all. SARS-CoV-2 came from nature. It's quite clear. It's ridiculous. Okay, so listen to this. The report also asserts that China knew even before the outbreak that the SARS-CoV-2 virus appeared to have severe, enduring neurological effects. No, it doesn't have severe, enduring neurological effects. In a fraction of people, long COVID has some neurological consequences, right? But there's no way the Chinese knew that before the outbreak, because you need to go a couple of months before you can say you have enduring neurological effects. This is such a stupid report by someone who knows nothing about infectious diseases. They think they further engineered it to be more neurovirulent. And he just cites all this nonsense. Kardec asserts that there's no evidence that the virus was deliberately released, but he says they may have weaponized it or something like that. They may have weaponized. So let's speculate. And he said maybe it got out. This is just such crap. It's unbelievable. So this is the part that gets me. The report's most contentious claim is that Chinese military scientists understood that the virus had acute and chronic short and long-term effects on the brain and may have been working not only to weaponize the virus, but also develop a vaccine. Where's that evidence? First of all, there's no way you can know chronic effects before the SARS-CoV-2 has spread around the world and has incubated for a while. You just don't know that. It's nonsense to speculate that. And they're going to weaponize it? What for? What are they going to weaponize it for? So they can kill their own population? This is ridiculous. Totally, totally ridiculous. And he cites some patent applications, tips, unpublished research, blah, blah, blah. But if he says it did come from nature, it didn't come from a lab, what is this all about? It makes zero sense. And yet it comes out, big splash, this guy Cadleck's article, and people say, oh, it was weaponized by the Chinese, huh? It must be right, because this expert on weapons of mass destruction, nobody has been able to weaponize a virus. The Soviet Union tried for many years. You have to lyophilize it, okay? You have to dry it out, because that's how you store it. And then you have to rehydrate it and make it infectious before you load it into the warhead. And you have a certain amount of time to do that. It just can't be done. People tried. A lot of people died in the Soviet Union trying to do that. It's absurd to think that they were weaponizing it. It just is ignorant, okay? I don't care what he's an expert in. He doesn't know anything about weaponizing viruses. Okay, that's that one. What else do we have here? Okay, more funding problems. Where is this? I have to find the article because it's in the New York Times and it's behind a paywall, so I had to download it here. Where's my folder? Okay, 2025. Oh, I already have this open. Why am I looking for it? It's already open. Here we go. We have 2 more articles to talk about. This is very concerning. This is an article in the Wall Street Journal, I believe. Yeah, it looks like it. Trump administration puts new chokehold on billions in health research funding. Okay? So basically, what they're doing is saying that NIH funding for the rest of the fiscal year can only go to NIH staff and salaries and expenses, not to new grants or certain grants that are up for renewal. So as you know, the vast majority of NIH funding goes to outside laboratories, the so-called extramural funding. They're saying now no more extramural funding for the rest of the fiscal year. Only stays at NIH. It's like $15 billion, almost half of the NIH budget. Well, the one I used to know, the new NIH budget, who knows? It's outrageous. It's outrageous. I just don't understand the justification. And they say the Office of Management and Budget is undertaking a review of NIH spending, some of which is now paused. Really? You just can't do this. You cannot just pause spending. People have to pay other people in their labs. If they can't pay them, they have to let them go. The whole lab grinds to a halt. And you think you can just start that up again overnight? Is obviously this budget person here, this guy, Russell Vought, doesn't know anything about how science works. You can't just pause. It's like, let's pause the airplane in mid-flight. Shut off the engines while we check something out here. Stupid. I can't believe this. Okay, one more story, because it gets me too riled up. So this is a New York Times study. Story by Christina Jewett. Top FDA official resigns under pressure. Vinay Prasad. So what happened? He quit, but they made him quit. And this is the pisser here. So this lady, Laura Loomer, is a right-wing influencer. These people know nothing also, right? And she writes columns saying that Prasad is really a Democrat. He supports Bernie Sanders, and he's made a few decisions they didn't like. So she wrote this op-ed in the Wall Street Journal, and it was sent to the administration, and then they put pressure on him to resign. But look at the smarmy nature of this comment by Andrew Nixon, who's a spokesman for HHS. Dr. Prasad did not want to be a distraction to FDA and has decided to return to California and spend more time with his family. If that isn't an insult, not that Prasad should not be insulted because he deserves it, because he insults everyone else, but that's a ridiculous insult to say he wants to go home and spend more time with his family, right? That's the typical excuse that everybody gives. Oh, I resigned to spend more time with my family, when it's really something else that they don't want to tell you. So why, Prasad, of course, doesn't say anything. Now, remember, Prasad, I never liked Prasad because from the beginning of COVID, he played it down. He kept saying that the cases were plummeting, and they were never plummeting because he doesn't know anything about infectious disease epidemiology. It was a good takedown by Debunk the Funk, okay, on him. So check that out over on YouTube. And, he recently says, no COVID vaccines for pregnant women and children. Myocarditis is too dangerous, when a report has just come out showing that pregnant women and children are at great risk for severe COVID. And plus, there's no more myocarditis, so I don't know where he's fabricating this from. So, if you're a fan of Prasad, that's all dumb stuff that he's doing there. And I don't think he was qualified at all to be head of CBER. But this is all full of just nonsense. Here, for example, this lady surfaced old social media post in which Prasad declared he was a political liberal, and another where he said he had a Trump voodoo doll. Really, you're bringing this up now? Why didn't you do your homework when he was nominated? He also cracked down on this company, Sarepta Therapeutics, that came under scrutiny after 2 teenagers and a 51-year-old man died after using the gene therapy that the company made for Duchenne muscular dystrophy. He reversed this decision a few days later for young boys who could still walk, but they didn't like that in the Trump administrations. Now, this guy, Senator Rick Santorum, another genius, has ties to Sarepta, the company that's making this adeno-associated virus gene therapy for Duchenne muscular dystrophy. And he was not happy with Prasad stopping the use of this therapy. Of course not, because he wants to make money from it. Here's the name of that Wall Street Journal article: a Bernie Sanders acolyte in Maha drag. She's an oncologist and an epidemiologist. So he made his name as an outspoken academic. Okay, so that's enough of that. I am not sad to see Prasad go. But I suspect he's going to be replaced with someone as bad or worse, because that's what this administration does. They replace, they use people who are not qualified in various positions. Every person in this administration, including the president, is unqualified for their position. So yes, Jill says debunk Dr. Dan Wilson. Yes, he is debunking Vinay Prasad. Someone should just debunk Laura Loomer, right? She writes all this nonsense. I mean, just nasty op-eds, just bloody nasty. But you know, if you disagree with someone, just lay out your opinions. You don't have to call them names. Ay. Okay, so let's see what we have here. Where's Vincent? I'm right here. I've been talking the whole time. I just was sharing the screen, so you didn't see me. I'm going to try to go back to where I left off. Okay, here we go. So, F. Yang, thank you for coming. I met F. Yang at ASV, Houston 33C. Ryman, dear teacher Vincent, thank you very much for your work and recommended readings and encouragement. I have a question. How do you deal with your local professor not believing you? Well, you sit down and have a quiet discussion of why the professor doesn't believe you, and you presenting evidence to say you should believe me. Okay, calmly, not yelling or screaming. Right? Yeah, that's the best way to do it. Rainey is in Raleigh, North Carolina, where it is 80F. Dottie says there's a big COVID surge here in California. Yes, here in the numbers. Last week's clinical update with Daniel, the numbers, the wastewater sampling showed a small uptick, and he said his cases were rising also. So maybe we're going to have another surge, another summer surge like we did last year. Leo says, I'm going to a retreat for 4 days. I'm the official morning COVID test monitor, rapid antigen test. They must know I'm a Twiver or maybe seen me playing a doctor on TV. It's probably they saw you on TV. Nobody knows about Twiv. Very few people. Dr. Racaniello needs an ad blocker. I presume so, but there's certain things I just don't pay attention to. I pay attention to what I think is important, and I'm sorry about the ads, but I just don't have the time to look into an ad blocker. Ryman, what do you think about the future of AI? Well, the AI is already here. It's used to analyze structures, for example. It's used to analyze big data sets, for sure. That's where it shines, in my opinion. Not in writing your papers for you, but many people tell me, people for whom English is not their primary language can get help from writing. Maybe that's true, to refine your grammar, but not to write the paper to begin with. That's not what the future is in virology. But analyzing data, big data sets, and structures, protein structures, for example, even, protein structures. I think that's where it's going to be useful. Vanity says there should be mandatory follow-up hearings to maintain their posts when they go against things they say in their initial hearings. Too many of them lie. They should have to account for what they do. Well, Senator Cassidy brought RFK Jr. back for a hearing, and nothing happened. So in this administration, Vanity, there's no accountability. Everybody's on Trump's side. The Supreme Court, the Congress, all the agency heads, they're all on his side. And nothing is going to happen except if they want it to happen. So we really need to break this. They've taken away all the checks and balances, all right, that are supposed to keep this republic properly functioning. But because of a combination of things, they destroyed them all. Visto, can I head HHS? I know nothing, so bound to do a better job than RFK Jr. Sure, I'll nominate you. You learned a lot here on these podcasts, right? Julie S. is in northern Pennsylvania. Hello, Julie. The weapon the Chinese used was not informing the rest of the world before it spread. They were a little late. They began to share information in December. And, the estimates are at the most it was starting to spread in late November. I don't think they were that bad at doing it. They could have been faster. But, sharing the sequence. Now, that's a very interesting story. The first genome sequence of the virus. There's maybe a guy in Shanghai, I forgot his name, and Eddie Holmes had to talk him into sending it to him. And he got in a lot of trouble for that because I don't know what the authorities wanted to do. Maybe they wanted to hold it for a while. That was not good. But fortunately, he released it and everybody got the sequence, and that's how they started to work on spike vaccines. City Journal may be a crank news source. It may be. But I just wanted to point out what they have this guy saying, right? As a way to back the story and to criticize it. I don't know anything about City Journal. Yeah. I think I heard on Twitter that patient 0 was identified. Which pandemic are we talking about here, Molly? HIV or COVID? Yeah, there is a very early patient who went to the hospital, but that not necessarily is patient 0, right? Nevitz, hello, all just sailing over from 3 hours of Hal Sparks. Put on my science hat. I don't know what Hal Sparks is. I guess it's something you watch for a long time. Rio writes, Judith Miller's 2001 book, Germs, has a strange reality. If you listen to the current audiobook, you will find it is heavily edited, has chapters out of order when compared to the original paperback. I don't know what that means. It's a fact. Okay. Hello, Aditya from Varanasi, India, 27C and pretty humid. I am actually baffled by these policies of RFK because we also have stupid legislators but they never question COVID shield vaccines. We also have stupid legislation. Oh, that's great. We are all baffled here in the U.S. about what is going on. But it's true. He's totally unqualified for his position. He knows nothing. And he thinks infectious diseases are caused by bad air, not by infection, not by viruses and bacteria and fungi. And he thinks that vaccines cause chronic diseases, so he wants to get rid of them. And Paul Offit thinks that aluminum adjuvants are the next of his target. So we'll see. It was another story I was going to bring in, but it has to do with cancer. You know, he fired the committee that advises healthcare companies about cancer treatments. So he's going to put his own people in that have verkakte ideas about this? It's ridiculous. Why? Every time he fires people, the reasons he gives are lies. Complete lies. Monday, listen to Paul Offit's Beyond the Noise about him taking money away from Gavi. Total lies for his reasons. Judith Miller was the fired New York Times reporter that said Saddam Hussein had weapons of mass destruction. The Times had to take pages and pages retracting what she wrote. A brown stick in the ground would be an improvement over the current HHS secretary. This is Project 2025. That's absolutely right. It's all Project 2025. Yes, Vinay is gone. It's good, but I just worry who they're going to replace him with. Nevitz says they're treating government science like a business. You can just, poof, pull the plug, move people around, fire people, poof. Yeah. They are malignantly ignorant. That's a good thing, right? Even though he was ousted by a loon, it is in my opinion, it's a good thing. I think he wasn't good. He wasn't qualified, and he didn't have the right background. So he's just not qualified. I was going to say something else. What else was I going to say? He had done things with respect to COVID that were totally wrong, and shouldn't have been appointed in that position. Molly says, my doctor added a new antihypertensive because my blood pressure keeps going up when I think about this stuff. I don't know if mine does. I don't measure it all the time. Peter says, Vincent, you have to realize that all the crap about lab leak and weaponizing of SARS-2 is all about increasing clicks and upping ad revenue for so-called news sites and Twitter accounts. Yes, I do. But why would this guy, an expert, a so-called expert on weapons of mass destruction, why would he write this document? He wants personal aggrandizement, right? That's a cool word, aggrandizement. It's for him to get more famous than he already is. Javier, 24C and cloudy in Ontario. Oh, it's November. I forgot. I'm going to Toronto in November for the ASTMH meeting, American Society for Tropical Medicine and Hygiene, which I attended before with Daniel and Dixon, and we did a TWIP and a clinical update, so we're going to do the same thing in Toronto. Jeff writes, Vogt is a Christian nationalist. That was the OMB, whatever, that pulled the NIH money, who has been working to destroy the government's capability to regulate in the public interest. He is an author of Project 2025. He and his ilk are the enemy. Yes. And you see, they think this country should be run in a certain way by certain people, and there should be no foreign influence, ignoring the fact that the way the U.S. has become successful is to have immigrants and to work with other countries. And somehow they have this verkakte notion that they are taking away all the jobs from Americans, which is just nonsense. You wait. Once all the immigrants are gone, you wait and see if any American picks up a hoe to go work in the fields where the immigrants used to work. Nope, they're not going to do it. They're going to say, it's just beyond, it's below me. I'm not going to do that. And then the farmers are going to be in trouble. They're not going to be able to harvest. Vista writes, science must align with the ideology of the administration or face defunding. So destructive to truth. Absolutely. Absolutely. Yes. Holy moly. Ties to the company. Santorum. Rick Santorum. If the Democrats were in charge, there would be hearings about this. Absolutely. Nobody cares about conflicts of interest from top to bottom. They all have conflicts. They're not going to get investigated. Yet, of course, the Democrats will get investigated for everything. Yeah. Questella says, I just looked up what Maha was because Questella doesn't follow the news. But, they also have this MAGA, right? Make America Germy again. That's what they're doing. It's not what you think it is. It's Make America Germy again. And I have to thank Noir for that, who emailed it to me some time ago. She said, I have this great thing, MAGA, Make America Germy again. I used it a couple of times. So I have to acknowledge Noir for sending me that. It's her idea. I wouldn't come up with that. Thank you very much. Lagotic, my feelings about Vinay have been mixed, but I think it's to his credit. He bailed on these loons. I think they forced him out, Lagotic. They don't like him, but I didn't him either. I'm not mixed about him at all. Nev, it's not only unqualified, they're anti-qualified. They're causing irreparable harm that will take years to revert. Trump's holdback of grants from Harvard and other high-level universities is nothing more than a mob-style protection racket. Plus, you may have read in the news that Columbia is paying a $200 million fine to the administration for all of their shortcomings. This is a racket. It's strong-arming, right? You don't fine a university. You sit down, you figure out how to fix the problem, and you move on. What is the point of fining that? It's a lot of money for a university, for example. Ridiculous. Joel, thank you for your contribution to science communication. Joel's in Bath, UK. I've almost finished my first research paper, Deriving Disease. You'll be able to read it soon if you wish. Thanks for your work. Whose job is it to collect wastewater samples? Well, as you know, wastewater in most communities gets processed through a plant, a wastewater treatment plant. And the people in the plant siphon off some of the water. They put it in bottles and they give it to the people who are doing the wastewater sampling. So it's already built into the system. Nobody has to go out and dip anything into the waste. John, thank you so much for your contribution to science communication. Another great week of content. Immune made me think of all the zoonosis on Noah's Ark. Is it zoonosis if a virus jumps from animal to animal or just to a human? Just to a human, because zoo, right, the animal to human, zoonosis. Animal to animal. What is there a word for that? I didn't ever hear of a word. No. Among animals, it's just transmission. Interesting, though. Jeff writes, regarding nasty conservatism is a cult of grievance and blame everyone for their bad decisions and failings. They attack experts because they seem as elites who don't respect them. Sad people. It's all right. You hope they get paid well in the waste treatment plants. I don't think they get particularly well paid. It's a job, right? College kid, I'm studying bioinformatics and would like to pursue virology as well. I've got 3 years left of my undergrad. Any advice for becoming an expert in your field? Well, you got a while to become an expert, right? But that's great. It's not likely that you have a virology lab at your undergrad, but if there is one, try to work in it. You got 3 years left. You have 3 years to work in a virology lab. Now, if there's no virology lab, what are you going to do? Is there one close by that you could work in? But research is the best way to become an expert. And then, of course, when you go to college, you can do it some more. You can work in a virology lab and take the courses you need, right? Organic chemistry, biochemistry, virology, if there is a course, and others as well. So that's what you need to do. And if you want to be a virologist as a career, if you want to be a PhD, you don't have to be a PhD. You can do it as a master's as well, but you can decide that when you're in college. Oh, sorry. Ryman said, sorry, I meant local professor not believing in you. Well, I don't give a damn if they don't believe in me. I base my opinions on science, and so the local professor is not. I mean, there are some things in science that you can have an opinion about if the data aren't clear. I think the data mean this. I think it means that. That's a valid discussion. But believing me, it's not about me. It's about the data. You don't have to believe me. You just believe the data. I tell you the data show this, and that's it. There's no questioning. Unless the professor thinks they know everything, and that's unfortunate. What do? The same thing. If you want to sit down with them and say, well, what's the problem? What's your problem? And here's why the professor is right. Me, meaning me as a professor. Tracy is in Santa Rosa, California, 75 degrees with a surge of COVID. AI is a great pattern detector, and so it sees things in data that humans miss. That's fine. Yes. Soul Symphony, well, Vincent, how much is known about SARS, long COVID in the brain? Okay. So there are long-term neurological consequences of SARS-CoV-2 infection. It's part of the long COVID syndrome, right? Brain fog, cognitive disorders, even some motor disorders, right? This is all real and people report them. What's causing them? In my opinion, no one has shown that the virus reproduces in the brain. What it does do, most likely, is induce inflammation elsewhere by reproducing, say, in the lungs, and the cytokines get into the brain and they cause damage, and that may be one of the reasons for long COVID neurological symptoms. And remember also, there are if you have severe pneumonia or let's say severe pneumonia after SARS-CoV-2 infection, you're going to be hypoxic, right? So that could be another source of the brain symptoms. But people know that these exist, but they don't know the cause. I'm speculating when I say these two things. Many people feel that the virus is reproducing in the brain and therefore causing these symptoms, but I don't see any good evidence for the production of infectious virus in the brain. The Funk interviewed Peter Daszak on COVID origins. Yeah, well, we did too a couple of times, Wild market origin, that's fine. And yeah, Peter Daszak appeared on TWIV a couple of times and had a problem with that because of the publicity he got. Hello, Jessica from the University of Toledo. It was cool to have Dr. Angela Rasmussen on. Yes, that was a lot of fun. Costello keeps forcing people to play Pandemic the board game. It's a good game, yeah. Jeff writes regarding follow-up hearings, like Vincent says, the GOP has made everything political. Their goal is to end proper regulation so that the market is unencumbered. Artemis, a friend has her mother in a nursing home and they just had an outbreak of human metapneumovirus. Is there anything of particular interest about this virus? I think it was under the radar for a long time. It was only discovered recently and added to another one of the numerous viruses that cause respiratory infections. And so I think that makes it of particular interest, that it's always been there and we didn't know it. Right? And now we have, we can diagnose it, obviously. Molly, thank you for your support of science communication. Really appreciate it. Thank you for your great shows. I watch as many as I can each week, at least 2 or 3. Thank you. Appreciate that very much. Hope you enjoy them. All COVID pandemic is what you were talking about. Yeah. I think I answered it in terms of COVID. Yeah. Questella, all the donators today are greatest of all time. Thank you. Thank you, donators, for supporting Microbe TV. Here you go. Zhang Yongzhen, the first person to publicly release the genome sequence of the SARS-CoV-2 virus. He got in great trouble for that. I don't know if he's still in trouble. But Eddie Holmes convinced him to do the right thing. And he got in trouble. He died of COVID. Is that right? Okay. Hello, Tona from Long Island. You don't have to tell us the weather because we all have it here in the Northeast. Julie says human metapneumovirus has been occurring in nursing homes for years. That's right. That's right. Dr. Shropshire, Bro Shropshire, welcome back from Birmingham, Alabama. Raining an 82F. Tom writes, I'd heard he lost his lab, hadn't heard of death. Could that be confused with the doc who went on social media about the new disease and subsequently died? I think that's right. Because this fellow, Zhang Yongzhen, yeah, he lost his lab, and I think he was even sleeping on the street outside of his lab at one point. We covered that story on Twiv some time ago. Yeah. I think he's still alive. Okay, let's see if I can translate this for you. Qu'on me donne 6 lignes écrites de la main du plus honnête, j'y trouverai de quoi le faire pendre. If someone gives me 6 lines written by the most honest man, I will find that I don't know what the last is. J'y trouverai de quoi le faire pendre. That is confusing me. So translate it for me. I got the first part. I will find it. I will find it something to think about, something like that. Pendre. Mark, I hope RFK really believes it. Great. Next time he needs surgery to not sterilize the medical instruments. You'll walk the Gowanus. All right. You can come. We'll find it a day when it's cooler. I will walk from the coast all the way inwards, and, we'll make some content along the way. Sure. It's a deal. Yep. Ryman, have you heard about the guy who allowed poisonous snakes to bite him for years and now developed immunity to most poisons? I haven't heard of that. Why did he live? He told them not to put too much venom in him each time. I haven't heard that. Hello, Margot from Phoenix Metro, 109F. Oh my gosh. Oh my gosh. Venomous, not poisonous. Okay, venomous. Thank you very much. If you ever come to Cincinnati, I have to know about it. I will let you know. I'll let everyone know where I'm going, okay? Because I'd love to meet, especially you folks who have been here for so long. It'd be fabulous to meet you. By the way, we're going to have a couple of fundraisers at the incubator this fall. Little intimate things. I figure local people will come. We'll have a little donation to get in. Maybe 20 people we could fit in, and then we'll do a live podcast. We're going to do Immune 100, which I think it's in December. Is that right? Yeah, I think it's in December. And then we're going to do something else before that. So just keep, I'll announce it here. But if you're in New York, obviously those are the people. There have to be 20 people who want to come to a live event, right? Kent, thank you for your contribution to science communication. Really appreciate it. You know, we do. The smallest to the biggest, we love it because it means you support the work that we do in getting science out there. Hopefully there won't be bad storms when you go to Toronto. Yeah, it was horrible getting to Montreal. Storms first there and then here in Newark. So yeah, November, I guess it would be snow, right? And Dr. Bro Shropshire congratulates Vanity for getting into medical school. Let's see. Yeah, hit the like button. We have 98 likes and 151 people present. And so just hit the like button so that we maybe can attract more people. Tom writes, a recent article in Chemistry, he got his lab back. That we're talking about the Chinese scientist who released the first SARS-CoV-2 genome sequence, but Eddie Holmes says he's been deeply affected. Okay, and the Wikipedia page for Zhang Yongzhen doesn't say he's dead, so I must have conflated him with someone else, right? Oh, okay, Eric translated. There you go. Pondra. If you give me 6 lines written by the hand of the most honest of men, I will find something in them which will hang him. Naive to think Ralph Barrick has nothing to fear. Yeah, this was Cardinal Richelieu. Thank you, Eric, for translating. Well, I got half of it with my crappy French. Molly, Elon Musk was the ultimate conflict of interest with all the contracts and doing away with agencies investigating him. Yeah, there's just no accountability in this administration. And I have a hard time believing that there are some Americans who like that. They get it. It's going to bite them in the ass at some point, like losing Medicaid. Jill writes, so grossed out to hear Rick Santorum's name again. I kind of thought we were done with that puritanical creep. No such luck, I guess. Costello writes, we have a septic tank. A lot of city stuff is foreign to me on some level, but we have trees. It's okay, Costello. Don't worry about it. Don't worry about it. Wait, can I take samples from a septic system? You could. Usually they're, just below the ground. There's a lid. You have to know where it is, and you make a little hole, and then you have to lift the lid up. It's kind of a pain, and it's going to smell really bad because I know when I was a kid, I grew up, my parents had a septic system, and it stunk because it was all messed up. It was all messed up. They didn't have a field, blah, blah, blah. Carol says, I once heard that Vina said he did not have time to listen to a bunch of virologists. Well, maybe he should. He only has time to listen to himself. He thinks he knows everything, right? He probably thinks he knows everything about kidneys, too. Oh, yes. Vanity sent me an article. Did you like that article I sent you about the brain stimulating the immune system when it spots sick people nearby? It's really interesting. I'm not surprised, right? It's very cool, though. Evolution works in really cool ways. Thank you for that. Yep. But we'll do that on one of our podcasts, I think. I guess with septic, the sample would be so small it would be useless. It depends what you're trying to do. If you want to know if someone in your family has things or what they have, it would be interesting to know. But yeah, you need a waste collection facility. If you want to know what's happening in the community, obviously you need a waste collection facility where everyone's toilets are going into that. Yeah. Thank you, Dr. Bro Shropshire, for your support of science communication. It's greatly appreciated and greatly needed. Hello, Will from Changsha, China, where it's probably really hot. You see, I now know where you're from. And last week you were somewhere else, Newcastle, I think it was. How do I remember this? Usually if you see something once, you forget it, like Newcastle. Questella writes, my kid listed his favorite things about school as breakfast, lunch, recess, science, and science fair. That's great. I guess the food is pretty good. Or just a break. Good. Maybe your son will be a scientist. How to become an expert. Work hard and harder. Spend thousands of hours studying and then even more hours. Yeah, Malcolm Gladwell. What does he think? It takes 30,000 hours to be an expert. I think I've hit that, right? I've been working on viruses for 44 years or something like that. But apparently I'm irrelevant, according to a prominent Rutgers professor who is a big lab leaker. Privyet, Veronika, warm summer night in Moscow. 20°C with clear skies. Changsha is 35°C and cloudy. It's damn hot. Yeah. Yep. Visto, could SARS-2 acute infection also trigger reactivation of other latent viruses that have neurological effects? Oh, yeah. I mean, it does induce Epstein-Barr virus, and that is correlated with long COVID. Yes. So that's another possibility. Herpes simplex viruses. Yeah, that could be part of it as well. That needs to be looked into. Good point. Good point. Evans writes, we lost the great Tom Lehrer this week. Musical genius, satirist, wrote the Elements song about many others. Yes, the Elements song. That was great. Love it. Flying back to China Monday, I watched Contagion on my phone. Got several errors. Good. Okay, here's another French version of that quote. Qu'on me donne 6 lignes écrites de la main du plus honnête homme, j'y trouverai de quoi le faire pendre. To hang. So pendre is hang. I got it confused with think. If I ever meet you in person, we have to play a round of Pandemic. Of course, you will. We will meet. We'll go to Iowa and meet, and maybe we can have a round of Pandemic with oh my gosh, the lady in Iowa. I forgot her name. It's terrible because I haven't used it in a while. It's on my phone. Hold on. I got it on my phone here. I'm sorry. On my phone, she just sent me. No, it's just her handle is Viruses Luna, which is what she uses here. Yeah, the three of us will play Pandemic. That would be cool. Tim Fried is the guy who gradually built up immunity to venomous snakes. Okay. This local will be there to visit the iguanas. Yeah, you'll still be there here in the fall. So we'll make it a trip. Yep. Look at it, Gowanus Walk. Yes, I'd be. Oh boy, people are interested in a Gowanus Walk. Today was pretty brutal. Oh my God, it was so hot. And there's so much construction going on. There's heavy industry and it's just spewing dust anywhere. There was a big metal recycling facility where they pick up metal and they crush it into blocks, and it generates all this dust. Oh my God. Well, that's just a part of the Gowanus. We went to Carroll Gardens section of Brooklyn, and so it was just around there. But I know if you go further inland, it's a little bit nicer. Betty's going to come too. Welcome, Angry Penguin. It's okay to be late. You can have your sunny beaches. Give me the Brooklyn waterfront. And the guy's blood, who allowed venomous snakes to bite him, is now used for research to develop antidotes. Cool. That's very cool. Costello writes, let me tell you, most of these hardcore Trumper people just say they do not have time to pay attention to anything. That's right. That's right. Okay, Richard, thank you. Someone has to translate. I can't copy this and paste it into a translator, so someone translate it for me. You mark the lid of your septic. Very good. So you could get a sample if you want. But you have to find someone to analyze it, right? Vincent, I fear for you. I hope you're cautious and safe and no one tries to hang you. I'm cautious. I go to the incubator every day. I just go back and forth. I don't know what else to do. I don't really go out and hang with a lot of people. So I don't know what else to do. But thank you. I appreciate the concern. Frankly, I don't think we're high profile enough. Our information trickles down away from us. So we have this, core of people who listen, then the information trickles from them. And it doesn't always get attributed to me. So it's probably okay. Hello, Lise. I'm Columbus, where it's hot and humid. And more on Costella's septic tank. It now has a plastic busted swimming pool on it, so I can never lose it. It has a leach field. Yeah, so basically, the septic tanks get full pretty quickly of water. And so then you put a pipe and you have this big area which you've dug and you put gravel in the bottom. It's 4 feet deep. And then you cover it and you put the pipes from the septic tank in it and they have holes in them. And then the overflow from the septic tank goes out into the field and it gets absorbed. And meanwhile, the solid stuff back in the septic tank goes to the bottom, is decomposed by microbes. So that's how it's supposed to work. And my parents never knew you needed a field. So for years, it was just a nightmare. It used to fill up all the time. Then you had to pay some guy to come and suck it. Oh my God, what a nightmare. Who wants to live in the suburbs with septic tanks? It's just barbaric. Now I have sewers. And in New York, I had sewers. Gail has a word, MAGA, Mothers Against Greg Abbott, Doge, Department of Grift and Extortion. And here we have another Russian quote. I don't know what it means. Molly, our septic system came to the surface, the tank and the drain field. Once I saw toilet paper in the backyard. Yeah, I used to see that in my parents too. It's gross just to think it's coming up. By the way, don't flush wipes into a septic system. That's going to really screw it up. Even though it says flushable wipes, don't do it. Don't do it. Logotic says, yeah, the reason my feelings are mixed about Vinay is because sometimes he seems utterly full of himself. Okay, so that's the bad part. So you think his science is okay? Vanity writes, did you see there's a GoFundMe for the national deficit created by the BBB? It's inconceivable that people will give money, though likely before they realize their personal loss resulting from the BBB. Big, beautiful bill. No, I wouldn't give money to this government. No way, even it's to do that. Costella's child wants to be a scientist. He's only 7 now, so hopefully I can have him not lose that hope. Yeah. Translated from Russian, I think it takes 10,000 hours to become an expert. Right. Rima. Right, Rima. Thank you very much. I heard RFK Jr. say that his son has long COVID. He said that during the testimony a couple of days ago. And what does he think? It's caused by the vaccine? Such a moron. Oh, that is Questella. That's cool. Questella's name in Russian. Very nice. Rima. Sorry, Rima. I used to talk to you a lot, and I remembered your name. I'm really sorry. Nevitt says that atop Mount Washington, New Hampshire. It will be 45 F, 7C. Wow, chilly. Molly, I will have to get the game for our camping trip. I'm staying in a hotel, but the kids are camping. This is very smart. We're going to Shenandoah. Good for you. Stay in a hotel and be comfortable. Rima. Got it, Rima. Rima. Most of the maggots claim they need to do more research. Sure, take away the NIH funds. That'll get you more research. Really good. Smart. Here we go. Zhang Yongzhen staged a sit-in/sleepover at the door of his closed lab. It's not like he was homeless and had nowhere else to sleep. Still under a roof and safe. Okay. Thank you very much. Appreciate it. Pandemic board game is not at all like Twister. It's an intense, mind-bending world. Ariozone writes, maybe a decade or so ago, the Gowanus Canal was given Superfund status. Okay, so they have still Superfund status, and the goal was to clean it up by 2025. But they're only 30% done, but they're still continuing the cleanup, but it's going to take a while. Yeah. We're not in the suburbs. Don't really have neighbors unless you count raccoons, opossums, and groundhogs and deer. Yeah, they're neighbors. They're probably nicer than human neighbors. Leo says septic tanks are tough to drive. That is true. That is true. Oops. The one time I don't read the chat, y'all start speaking Russian. That's very funny. I hear Gowanus, and as a former New Yorker, I reflexively pinch my nose. Yeah, it wasn't so bad. It wasn't so bad. I didn't really smell anything. There was a strong wind, probably in the wrong direction today. Okay, everybody loves Pandemic, apparently. Marge is in Northern Virginia and Maryland, where it's 30°C. Richard is from Brighton Beach, has a lot of Russians. Yeah, that's the Russian community in New York. In fact, the Oceana Theater does performances in Russian. Diazone. Hello, dear all. Postdoc in Mount Sinai. I'm PhD from Japan, Kanazawa University in Virology, from nice but smaller, peaceful city. Center of Manhattan. Sounds fun but scary a little. You're thinking of doing a postdoc at Mount Sinai? Yeah, New York. You'll get used to New York. New York is a lot of fun. Yeah, don't worry about it. Don't be scared. It's good to be scared. You'll get used to it. Okay, I'm going to do a mini lecture now. It's 9:18. This is going to take about 20 minutes, I think. And let's get it started here. Oh, I have to take off that last comment. Let me go there. Yes, Diazone from Kanazawa. Cool. Okay. This is reverse transcription and integration, part 2. So remember, what happens with retroviruses? They are viruses with an RNA genome. The genome enters the cell. It's converted to a double-stranded DNA by reverse transcriptase enzyme, which is present in the particle. It enters the nucleus where it then integrates into the host cell genome. We call that a provirus. And from there, mRNAs are made that eventually specify the production of new virus particles. So this integration part is what we're going to be talking about now for the next couple of slides. The provirus is a permanent part of the host genome. There's no way to get it out. And that's why HIV infection is a problem, because the provirus is present in memory T cells that live a really, really long time. And you can't get rid of it. And it's silent. The only way the virus is made is by transcription by host RNA polymerase II. So we saw in that previous slide, the provirus DNA is transcribed by host polymerase II to make mRNAs, and that's the only way that you can make new viruses. Now, this provirus can happen in somatic cells, right? It could happen in muscle cells or heart cells or liver cells. And those cells eventually are going to turn over. They're not going to live forever, and they don't contribute to your offspring. So if you have proviruses in your somatic cells, they're going to stay with you when you die. But if the provirus is in germ cells, then you're going to give it to your offspring, right? Because offspring gets DNA from male and female parents. And so that's called endogenization, when the retrovirus integrates into the germline and it gets passed along to offspring. So the genomes of most organisms are full of ancient and modern retro elements, that is, things that move around by reverse transcription, including endogenous retroviruses. So, for example, here we have a population of people, and in this population, one person got infected with a retrovirus, and it went into their germline. And then, these people have offspring, and so that founder with the endogenous retroviruses passes on to other people, and eventually the number of people in the population that have the endogenous retrovirus grows. It just takes thousands and thousands of years to this to make a difference. Now, as the population grows, mutations get fixed in our DNA because we make mutations all the time, and most of the time it's not a problem. Sometimes it causes problems of diseases of various sorts, like cancers. But sometimes these mutations fall into the endogenous retrovirus, the provirus, and it makes it no longer able to make infectious virus. And so that's what we have here. These light red people are no longer making infectious retroviruses, whereas the dark red people are. And eventually, with time, in some organisms, you can have all the proviruses mutated so that no infectious virus is made. And you could also lose it early on, just randomly by DNA recombination. But right now, humans have lots of endogenous retroviruses. I'll show you in a minute. Now, this process of endogenization, that is, the provirus is in germ cells, and then it gets passed on to offspring. In humans, it happened many, many tens of thousands, 100,000 of years ago, before Homo sapiens even. But so we don't really know how it happens. But recently, koalas are getting endogenized, right? So koalas are native to Australia. They're only existing there unless they're sent to a zoo somewhere else. They only live natively in Australia. And about 50,000 years ago, they picked up a retrovirus from rodents, which went into their germline. And we can see the population of koalas in Australia being endogenized. In other words, we can watch these integrated retroviruses, that are now called koala retroviruses, spreading in the koala population. And the goal is, of course, to identify some koalas that are not yet endogenized and keep them for breeding purposes, right? Because they don't have it's very hard to get the retrovirus out. So here's a map of Australia, the eastern portion, and different areas where they have sampled the koalas and what fraction have the koala retrovirus in the germline. You can see in the north here, every individual, these are 27 koalas were tested and 27 were positive. And then as you move south, you have increasing numbers of koala retrovirus negative koalas. And in this island, Phillip Island, none of the koalas were positive, although some other islands, Kangaroo Island and Snake Island, Raymond Island, etc., these all have a fraction. So somehow the koalas got there, and they're breeding, and the retrovirus, the endogenized retrovirus, is spreading in the population. So we can actually study this endogenization in real time, and people are doing that. And not too long ago on TWIV, we did a paper where they are studying the components of the koala that contribute to this endogenization. So it's a really good opportunity to study endogenization, because in all other species, as far as we know, it happened so long ago. By the way, this endogenization, it makes infectious virus, and it's immunosuppressing the koalas. So they're getting all kinds of infections that are threatening their existence, and people are really working very hard to avoid that. And that's why we take Corv koala retrovirus negative koalas and make a breeding colony out of them. Okay. Now, retroelement is the name we give to sequences in DNA that move around the genome via reverse transcription. And this includes proviral DNA integrated in the germline endogenous retroviruses that I've just been talking about. And I've also told you they're often replication defective. In fact, in humans, they're all the endogenous retrovirus elements that we have are replication defective. They don't make infectious virus. They do make perhaps particles, but not infectious viruses. But wild mice, for example, do make infectious retroviruses from their endogenous retroviruses. So how do other retroelements move around the genome? Well, for example, here we have a gene being transcribed. This happens to be a retrotransposon. It's an element that can move around. It's transcribed into RNA. That's copied into DNA by reverse transcription. There's reverse transcription in the nuclease, and then it inserts somewhere else. So that's what we mean by sequences that move in the genome via reverse transcriptase. We go from RNA to DNA and we integrate somewhere else. 42% of our genome is made up of mobile genetic elements, including endogenous proviruses and other kinds of retroelements, and we're going to go over that right now. So 42%, that's 2.7 million copies of various retroelements. Okay, so on the right are the numbers, and on the left are descriptions of what they look like. So let's just go through that a little bit. Here we have endogenous retroviruses, which we've talked about, right? They have the retroviral genome with LTRs, and they have the host DNA duplicated on either end. And we talked about that last time, how that happens. So that is an endogenous retrovirus. And these constitute about 8% of the human genome, endogenous retroviral sequences. So they contain LTRs, and here are complete retroviral genomes. Then we have an element called a retrotransposon, which it looks exactly like an endogenous retrovirus, but it's missing an envelope gene, which it's basically the spike protein of retroviruses, right? Now, we think these are the precursors of retroviruses. We think retrotransposons existed in cells long before retroviruses, and to make a retrovirus, this element captured an envelope gene from the cell, and that allowed it to leave the cell and become a bona fide virus. But we have 0.6% of our genome as retroelements, retrotransposons, these guys here. Then we have what are called line elements, long interspersed nuclear elements, which have no LTRs, but they have open reading frames, and they have a duplicated sequence of the genome on both sides. So it suggests that this went in by integrase. But these constitute over 20% of the human genome. You can see here, they're non-LTR retroelements, and they can move around. They can move around. In fact, one of these open reading frames is a reverse transcriptase. Then we have what are called processed pseudogenes and signs, short interspersed nuclear elements. These have no reverse transcriptase. They do have repeated cellular sequences at either end, suggesting an integrase integrating mechanism. And these are basically mRNAs that have been converted to DNA and then integrate somewhere else. And that was the process that I had illustrated right here in the previous slide. And so these signs and process pseudogenes are, about 14% of our genome. And of all of these, less than 0.05% are active in the human genome today, which means they're being transcribed. So these are all the retro elements. 42%, almost half of our genome, consists of sequences that move around. It's really amazing. Okay. Now, sometimes, if you've got a retroviral gene in their genome, a provirus, sometimes pieces of it are used because we're talking about hundreds of thousands of years of evolutionary time. The cell may say, I could use this gene. Let's use it. And that's called exaptation. And here's an example of an exapted retroviral envelope protein, which was exapted to provide the placenta in mammals. So remember, the envelope of retroviruses binds the receptor on a host cell, which is shown here, and then catalyzes fusion of the virus and the cell membrane. So the envelope is a fusogenic protein, like spike. Many millions of years ago, like 50 million and 25 million years ago, on two separate occasions, an organism has stolen an envelope gene from an endogenous retrovirus and turned it into a protein called syncytium, which catalyzes cell-to-cell fusion. And syncytium is essential for the formation of the outer layer of the placenta. So here's the placental structure here with the fetal villi and the uterus here. And this red outer layer is called the syncytiotrophoblasts. It's actually one giant fused cell. There are no cell walls here. There are no membranes, many nuclei, because the cells fuse because they produce syncytium, which is none other than a retroviral envelope protein. Without syncytium, we would not have placentas. Okay, we'd be laying eggs or having live birth in the animal going into a pouch or something like that for a number of months, like kangaroos. So in this sense, this retroviral infection was essential for mammalian live birth development. So here's another one. There's a retrovirus called HERV-K, which infected our ancestors about 200,000 years ago. And that would be around here. So, it probably infected Neanderthals or Rhodensiensis and passed on to us. We still have HERV-K. They don't make infectious virus, but mRNAs are produced during normal human embryogenesis from the 8-cell stage all the way to the epiblast. So remember, in early development, you have an oocyte, which is fertilized, then it begins to divide, becomes 2 and 4 and 8 cells, a morula, and eventually a blastocyst. And the HERV-K mRNAs are produced from the 8-cell stage through the morula to the epiblast. Then it stops. And you can actually see virus-like particles in their cells, but they're not infectious. The presence of these retroviral particles induces an antiviral protein called IFIT-M1, and that's thought to be important for the defense of the developing embryo against virus infection. So somehow we have exapted HERV-K to help provide embryonic defense, right? It's very cool. And one more example of beneficial retroviruses. You've seen white chicken eggs and brown chicken eggs and blue ones. The blue ones are highly prized, and farmers breed to get blue eggs, to get chickens that lay blue eggs. The reason they're blue is because a retrovirus has integrated next to the gene that makes the blue pigment randomly, because chickens have lots of retroviruses in them. They're actually infectious retroviruses in chickens. We talked about that before. And if it randomly inserts next to the blue pigment gene, you get blue eggs. And you could breed, the farmers can breed these chickens so that they have a pure line of chickens that make blue eggs. So without retroviruses, folks, we'd be laying eggs and they would be white. Okay, the last topic I want to talk about is the hepadnaviruses. These are unusual because they are DNA viruses that encode reverse transcriptase. Here's the hepatitis B virus particle. There is an electron micrograph there on the left that has an envelope which contains viral glycoproteins, and then within it is a nucleocapsid that has a double-stranded DNA. This DNA is actually partially double-stranded, has some single strand, it has a piece of RNA attached, it has a protein attached, and this DNA is going to be reverse transcribed at some point. So looking back at the Baltimore scheme, if you remember this from eons ago, this is showing how the 7 classes of viral genomes make mRNA, right? We have our double-stranded RNA, our minus RNA, our plus RNA. We have our plus RNA with reverse transcription. And then we have the group 7, which Baltimore didn't know about back in the 70s when he made this scheme, where you have double-stranded DNA, which is transcribed to RNA, which is then reverse transcribed to DNA to produce double-stranded DNA. So let's look at how that works. So this is a unique 7th family member in the Baltimore scheme. So the Hep B virus fuses to the outer plasma membrane of the cell. The nucleocapsid is deposited in the cytoplasm. It already has double-stranded DNA in it. The double-stranded DNA goes in the nucleus. It's not suitable for transcription yet because it's single-stranded, partially single-stranded. It has a protein and an RNA linked to it. That all gets fixed by nuclear enzymes so that we have covalently close circular DNA. And that can now be transcribed by host RNA polymerase II to get small RNAs that make viral proteins, structural proteins, and a large pre-genomic RNA. The pre-genome gets exported into the cytosol. It's packaged into capsids. Now, as soon as the capsid is made and wrapped around the RNA, what's also included in the capsid is a molecule of reverse transcriptase. And that begins to copy the RNA right away. It can't wait to get into the next cell like the retroviruses do. So then you end up having new hepatitis B virus particles produced that have DNA in them and not RNA. So that's why this reproduction cycle is unusual. And furthermore, the genome does not integrate into the cells, although it does remain as a covalently closed circular DNA. And if this cell lives, that DNA will remain in the cell. Now, I want to go over the reverse transcription. Last time we talked about it for retroviruses, but I want to tell you how we get this weird hepatitis B virus, partially double-stranded DNA with a protein and an RNA piece attached to it. That's going to all come out of this slide. So here's the RNA. Remember, we made RNA in the nucleus. It goes out into the cytoplasm. It's packaged in a capsid along with reverse transcriptase, this light brown protein, and another protein called terminal protein. Reverse transcription then initiates at this stem loop structure. But after a few bases, that product flips to the end, the 3' end of the genome. So you can see it flips here. It doesn't keep going to the end here as it happens in retroviral RT. It flips to the 3' end right away here, and then the reverse transcriptase keeps copying it. There's an RNase H that removes the RNA template. You end up with a full-length DNA copy of the viral RNA with reverse transcriptase still attached to the 5' end. There's a little piece of RNA left at the end of the genome, and that is a primer for the second strand. So you eventually make the second strand. There's another template jump here. But what we end up doing is making a lot of the second strand, but we never finish because this is happening inside a closed capsid, and there are not enough nucleotides to finish reverse transcription. So what we end up with is a partially double-stranded DNA molecule. The reverse transcriptase is still attached to the 5' end. You can see the blue ball there and the tan ball right there. That's reverse transcriptase. And then there's a piece of RNA. So there's an RNA primer still left on the genome, and that's shown right there. See, it's covalently attached to the genome. And all that has to be replaced and made fully double-stranded before transcription can occur. So it explains why the genome looks like that. And so we have a number of different kinds of viruses with reverse transcription in their reproduction cycles. So, for example, we have the retroviruses, and the genome in the box, the yellow box, is what is packaged into virus particles. So the retroviruses package RNA, and when they infect the cell, the RNA is converted to a double-stranded DNA, which is then integrated, right? RNA in the particle. The hepatinoviruses have DNA in the particle, which is released into the cell. It's repaired and then transcribed to pre-genome RNA. And then the pre-genome RNA is packaged, but then it's copied immediately to double-stranded DNA. So what ends up in the virus particle is actually a DNA genome. There are some plant retroviruses called para-retroviruses. So one example is cauliflower mosaic virus. These also package DNA, as do the hepatitis B viruses. And again, the DNA is released into the cell, RNA is made, it's packaged, but then the virus particle, reverse transcription occurs in the virus particle even before it leaves the cell. And then finally, we have the foamy retroviruses, where DNA is packaged in 5 to 20% of the particles. So not RNA like the retroviruses. These are a different kind of retrovirus, but they package DNA, probably by the same mechanism as we've talked about for Hep B and cauliflower mosaic virus. So that ends it for reverse transcription and integration. I do think we have some other lectures to do, so we'll continue those. And since you all liked having a guest, we'll start having guests maybe once a month, and maybe our next guest will be Dr. Daniel Griffin. Okay, back to the questions. Oh, sorry, Vincent, I assumed you spoke Russian. No, I'm sorry. I just know a few words because of the people that are here. Everyone get your notebooks out. Oh, I bought a new notebook, which is very cool. Look. So one of our listeners turned me on to these field notes, right? These are very cool. They're little notebooks that you can fit in your pocket. Neat. You can write stuff on. But then they also make these spiral bound ones. They're the same size. It'll fit in your pocket. And the top flips up. So I'm going to use these for interviews. I don't want to sit on stage with an interviewee for TWIV with a laptop anymore. It's too cumbersome. I want to have a little notepad like this with my questions on it. Anyway, why did I say that? Get your notebooks out. Yeah. How does a DNA sequencer work? Oh, that's a good question. Mini lecture next week. How's that? Because there are many different technologies, or several different technologies, okay? And I would love to compare and contrast, starting with the technology I used to sequence the genome of poliovirus. So what a great thing to do. Let me write it down. And so mini lecture for next time, DNA sequencing technologies. DNA Seq Tech. Thank you. Thank you, Angry Penguin, for that suggestion. Heather is in Kamloops. Welcome. 37C. Richard says someone from Russia told Brighton Beach reminded him of the Soviet Union because a lot of people escaped in 91 to come here. Now, herpes is not a provirus. Herpes remains extra chromosomal in the cell. Could RNA interference disrupt at the provirus stage? Now, RNA interference will only disrupt RNA, so it would disrupt the transcribed RNA, but the provirus remains. Could you remove it with CRISPR? Yes, you could. But CRISPR has off-target effects, so that's probably not going to work. But there are other technologies that have fewer off-target effects that are analogous to CRISPR, and they may. But that's the idea for removing all the HIV proviruses, right? I heard one issue of HIV is that it affects half chromosome cells, like sperm and ovaries. No, it doesn't. It doesn't infect them. It infects largely T lymphocytes of the CD4 variety, and they become memory cells and last a long, long time. Questella, I'm going to try to redo the beginning of the course when the kid goes back to school and hopefully can follow a lot more. And you might find the book useful also, the virology book, right? John wants to call the first infected retrovirus person Dave Thomas. It should be illegal to take an animal from its native lands, like taking koalas, huh? Well, good luck with that one. We should walk the Gowanus, then stop into the Bell House where Vincent could do a moth story slam. Yeah, I have an assistant who does moth stories in Brooklyn too, yeah. Koalas are quite inbred due to excessive logging of their food trees. Sadly, my tax dollars used to hurt our koalas. Molly went to a koala preserve near Brisbane last February. They are so cute with fuzzy ears. People are not really very kind overall. Well, there are kind people. Maybe a third of the world's population are not very kind. And a third don't do anything about it. And the other third are the ones who are the receipt of the non-kindness. Good question. For primary integration into nuclear DNA, must the DNA be in histone's unwinded state? Often it's in actively transcribed DNA, which is unwound, right? Did you see my stand-up routine? It's okay. I wouldn't say it's brilliant, but thank you very much. Can they make infectious retroviruses if a co-infection with an envelope from another retrovirus took place? What do you refer to by they? The retroelements or the endogenous retroviruses? The endogenous retroviruses in us have too many mutations, and so just envelope will not do it. But I think you're talking about retroelements. Yes, in theory, if the retroelement genome is intact, the open reading frames are intact. But you need gag protein, and I'm not sure that they have that. So I don't think that would work. Yeah. I've never been to the Bell House, and so we'd have to do a timing because, they don't let just anyone do it, I guess. I don't know how it works. Could transposons Visto asks, sometimes be a positive evolutionary mechanism for the species? Totally. By moving genes around and creating new genes, definitely has been a positive evolutionary mechanism, but has also killed some organism, but that's a dead end, right? And that's the end of it. Some human diseases are known to be caused by transposons, for sure, moving around, interrupting a gene and causing a problem. John says the human genome resembles an oft re-recorded cassette tape from my youth. That's great. I really like that. Good job. Good job. As in RSV. Let's see what. RSV, Rouse sarcoma virus. Yeah, that's a retrovirus that is going to integrate. HIV integrates. Every woman in labor, curse that ancient placenta retrovirus. Yes. We could always have a C-section, I guess. Marsupials don't even notice giving birth to the little jelly bean Joey. Yeah, the little kangaroos, that's right. They're so small, they don't hurt, and there's no placenta because they're not placentals, right? They weren't infected by retrovirus. And the little jelly bean size crawl into the pouch, and there they develop. Yeah, it's very, very cool. One has to remember that the capsid is three-dimensional. It's an envelope sphere. The picture just shows a 2D cross-section. That's right. That's right. Placental mammals have existed for 210 million years. Presumably other proteins have been adapted for placental functions. No, the placental syncytians are exapted from retroviruses. And I don't know about the number, Tom. Let me go back to my slide. I had a phylogenetic tree there. Here. So this one has 60. Well, I got to share it with you. Sorry. This one has 60 million years, right? And we have one transfer at 50 million years, and that is shared by prosimians, New World monkeys, Old World monkeys. This is just the primates, okay? So yes, you're probably right that there are other proteins that were exapted other than retroviruses for those other non-primate animals. That's right. Kang wants to know if there are retrovirus sequences in chimps and humans that have matching sequences. No, they're related but not matching because there's so much evolution in between that you have a lot of mutagenesis, right? So you can see relationships, but even in humans, there are a lot of endogenous retrovirus. So we make phylogenetic trees of them, and some can clearly be supposed to be derived from some ancestor. Well, remember, chimps and humans, chimps didn't become humans. They have a common ancestor, right? So chimps and humans share something that looks very similar, which presumably had a common ancestor, had an origin in a common ancestor. Yeah. The inside of a brown egg is white with blue eggshells. The color goes all the way through. If you cross a brown egg laying breed with a blue layer, you get olive egg sheaths. Thank you, Shells. Thank you for that explanation, Red Hen. I appreciate it. And so the blue pigment is turned on. The gene is turned on by a retrovirus integration. Melinda wants to know, are there solutions for post-polio? No. There aren't any, unfortunately, because you're losing motor neurons usually as you age. And then if you've already lost a lot to polio, then you don't have any backups to compensate. And the only thing you can do is try therapy, but many individuals don't respond and they end up in wheelchairs. Yeah. Olive or green eggshells are blue on the inside. The brown is added as the eggs move through the oviduct. Oh man, Lagotic says, I want to start a new career breeding colored eggs. Never heard of them before. Why does COVID-19 trigger tachycardia in many people? And what resolves it? I don't think we know. I think it may be. So in hypoxic individuals who, they can't get enough oxygen in their blood, that can trigger it. So in that case, oxygen would be the solution. And that often resolves it, right? And in fact, even in people who have tachycardia without COVID, eventually it will resolve due to oxygenation. So I think that's one possibility, and there may be others as well. Oh, so everyone likes guests, and they will look forward to Daniel. Okay, so we'll do that. Visto wishes he understood more. Ask questions. You ask a lot already. It's okay, but ask more. Tona wants to learn about Dr. Griffin's hobbies like his sailboarding. Just ask him. He'd love to talk about sailboarding. Absolutely. Anything. He's a very good at talking about everything. Molly says moleskin notebooks are the same size. Yes, they come in different. These are much cheaper than moleskins, I can tell you. One moleskin is like $14, and those are much cheaper. I don't have a lot of money. Man, we will need tech cursive writing to use the old-fashioned notepads. Mini lecture on modern DNA sequencing. Okay, I'm going to do it. I'm going to have to prepare extensively for that, but I'd love to do it. Yeah. Yes, I'm going to start with Maxim and Gilbert, and then we'll do Sanger, and then 454, which was the first next generation sequencing, and all the way up to the present. Lavishly illustrated. I'll steal illustrations from the net. Logotic says, if I ever wake up in a New York hospital, I'll feel better if Dr. Christian shows up. Well, he's just going to, he'd be at Columbia, okay? He'd be an ID consult. So if you get admitted to Columbia Presbyterian, otherwise you have to be out in Long Island. Red Hen has had hens that lay white, brown, green, pale pink, and blue eggs. As PCR drastically evolved in the last 25 years. Yes, it has. I don't know. Basically, multiplexing and the addition of dyes so that you don't need to use radioactivity and some other things. But there have been other approaches to amplification. So polymerase chain reaction is a certain kind of amplification, but then there are another nucleic acid amplification, and NAT can be done with other enzymes and by other approaches. That's another topic for a mini lecture. Another one is PCR evolution. Let me write that down too. That's good. PCR evolution. Cool. Melinda asks, what nutrient deficiencies are triggered by virus infections? I don't actually know. All I know is that many viruses utilize glucose extensively, so they induce breakdown of glycogen and other carbs to get glucose. They also induce the synthesis of lipids. But in terms of nutrient deficiencies, I don't know. I'd have to look into that. It's a good question. Variety pack of highlighters. I love highlighters. I got to tell you, I am a pen and pencil freak. I don't use them as much as I used to, but I have tons of containers like this. This is a TWIV mug and all kinds of pens and pencils. Of course, the much needed scissors. But I mean, look at this one. I got this at some conference. Look at how unusual. It's just a clicker, but it's an unusual top. I love pens. This one is interesting. It's from Japan. It's got a little teddy bear or something on it. Anyway, I got lots of these because I had a pen fetish for the longest time. I have this one, which is cool. This has got a piece of circuit board embedded into it, but you don't want to know about that. Okay, sorry. It would be great to have Amy on, even if she talks about what she has clearance to talk about. The only hope for koala survival may be the colony in California. UK, us Aussies not look after them well. Racism is a virus of the mind. Racism and there's other things I'm forgetting. Noir, thank you for your support of science communication, requesting Daniel Griffin. Okay, we got to wrap up here. Let's see what else. Tona was in Sydney and paid extra to hold a koala. Now that I think back, that's not cool. Yeah, well, they're captive, right? Do any other types of organisms incorporate into our DNA? There's some bacterial DNA, and of course, our mitochondria come from alpha proteobacteria. So, yeah. Wikipedia says 105 to 120 million for placental mammals. Let's see, what else here? Ariozone's happy when I recognize Gag and Paul on my slides. Lots of comments here, but I'm going to skip to the end just to see if there's anyone who has donated. Yeah, pens and markers must breed naturally in a coffee mug medium. That's absolutely right. Yeah, Japan has great pens. When I went years ago, I bought some great pens there. Oh my gosh, they're really good. All right, that'll do it for another office hours. We have good number of likes. That's great. I want to thank our moderators. We had Les, we had Vanity, we had Steph SF, and Tom Steinberg. Thank you so much for moderating and keeping it civil. Thanks to everyone for coming. And as Lagotic said, we must work to make the Gowanus Canal Walk happen. Yeah, but we're talking about the fall when it's cooled. September, maybe October even. September can be warm, but it's going to be a lot better than it is now. But we'll talk about it here and give you ways to communicate before that happens. We're going to be back next week. Let's see. I'll pull open my calendar here. Next week is the 6th of August. We'll be back at 8 p.m. Eastern for another office hours. I love you guys. You're great. Thank you so much for being here, and we'll see you next week. Meanwhile, please be safe. Bye-bye.